Welcome back to this next video in which we are discussing the control of blood pressure and blood volume. Okay, so we are currently discussing the baroreceptor response. So, where have we got to so far? So, remember, we have seen that the information about blood pressure is going to be conveyed to the nucleus tractus solitarius on both sides. What I am trying to explain to you at the moment is the different centres in the medulla for controlling sympathetic output to the vasculature and sympathetic and parasympathetic output to the heart. So far what we have seen is the vasomotor centre, which controls sympathetic output to the vascular smooth muscle cells surrounding arterioles, specifically or particularly in areas such as the skin, the skeletal muscle and the gastrointestinal tract, where we can alter blood flow there. For instance, we can reduce blood flow there and not have to worry too much, whereas areas like the brain, for instance, we don't want sympathetic nervous system being able to alter the blood perfusion of those areas. Right, so the vasomotor centre, remember, it's this cluster of neurons, or this network of neurons, inside the medullary reticular formation, and they're all going to be talking to each other, and then the means by which this actually has an effect on the sympathetic output to the vascular smooth muscle cells is that you have these output neurons, these preganglionic sympathetic neurons, if you like, which are going to descend down the spinal cord and synapse onto the preganglionic sympathetic nervous system neurons that are going to be involved in stimulating vascular smooth muscle cells. So these then are going to leave the spinal cord via the ventral root here, and of course this will have its cell body in the lateral horn of a portion of the spinal cord between T1 and L2, and it will send out its axon in the ventral root of one of the spinal nerves at this level. And then its axon will end up going into the sympathetic chain. It'll synapse onto a postganglionic sympathetic neuron, which will be in one of the sympathetic ganglia, and then that postganglionic sympathetic neuron, its axon will return into one of the mixed spinal nerves, and then it will go off and find the vascular smooth muscle cell that it's going to innervate and release noradrenaline onto that vascular smooth muscle cell. Well, most likely it'll release noradrenaline onto lots of vascular smooth muscle cells, and hence control how much sympathetic stimulation we are getting to vascular smooth muscle cells surrounding these arterioles in these areas, and therefore the contracted state or vasoconstricted state of those arterioles. Right, now before we actually look at how the nucleus tractus solitarius is going to influence the vasomotor centre, let me say something about the sympathetic control centre for stimulation to the heart, and also the control centre for parasympathetic output to the heart as well, which are up in the medulla. Okay, so it's exactly the same principle as for the vasomotor centre in the case of this control centre for sympathetic output to the heart, which is why this is going to be a much shorter discussion than was the vasomotor centre. So there is a centre in the medulla known as the Cardioinhibitory Centre, okay, which for short we will abbreviate down to the CAC. So C for cardio, A for acceleratory, and then the second C is centre. And this is again in the medullary reticular formation. So again, it's a diffuse network of neurons rather than a cluster of neurons that we can say it's there. Instead, it's not localized to one specific point. It's a cluster of neurons that are all talking to each other in the medullary reticular formation. And just like the vasomotor centre, it's going to be determining how much sympathetic stimulation we are going to be delivering to the heart. And the means by which you'll actually get the Cardioinhibitory Centre communicating with the preganglionic sympathetic nervous system neurons is again from the Cardioinhibitory Centre. You'll have outward projections which will project down to a level of the spinal cord between T1 and L2 and will synapse onto preganglionic sympathetic nervous system neurons. Okay, so this picture that I've got here, it could equally well be the Cardioinhibitory Centre that I've drawn there as it could be the vasomotor center. So the picture is exactly the same, and it will come down onto preganglionic sympathetic nervous system neurons, which will then synapse onto postganglionic sympathetic nervous system neurons, and those postganglionic sympathetic nervous system neurons, their axons will join nerves, peripheral nerves again, and then will make their way to the heart, in this case to release noradrenaline onto the heart, which is then going to raise heart rate and also increase the force of contractility of the heart. Okay, so remember I mentioned about how the postganglionic sympathetic nervous system neurons that are going to innervate the heart, they mainly come from the cervical ganglia, so they mainly have their cell bodies in the cervical ganglia and the upper thoracic ganglia. So I mentioned that earlier on in my discussion of the vasomotor center, so I'll just come back to that now. Okay, so the postganglionic sympathetic neurons that actually are going to synapse onto the heart and release noradrenaline onto the heart, they're going to have their cell bodies in the cervical ganglia majorly and the upper thoracic ganglia. Okay, so that's the Cardioacceleratory Centre, so just like the vasomotor centre, it's going to determine how much sympathetic outflow you've got to the heart. Okay, finally what I want to talk about before I put this all together is the Cardioinhibitory Centre. So Cardioinhibitory Centre this time, which is usually abbreviated down to the CIC. Now this is the name for the control centre for parasympathetic stimulation to the heart. And this consists of the nucleus ambiguous, portions of the nucleus ambiguous, and also portions of the dorsal vagal nucleus. And this is going to have the preganglionic parasympathetic neurons, cell bodies in there. Okay, so I'm going to, where should I put this? I could try putting it on this picture. I'm not actually, I'm going to go down here and do it down here, so I'll just make sure this is straight. Excellent. So if I've got a picture then of my dorsal vagal nucleus here, but it could equally well be the nucleus ambiguous. So I've zoomed in on the dorsal vagal nucleus, but as I say, equally well it could be the nucleus ambiguous. And when we talk about the Cardioinhibitory Centre, the CIC, we mean the cell bodies of the preganglionic parasympathetic neurons that are going to go to the heart, which are located in the nucleus ambiguous and also the dorsal vagal nucleus. So let's say I have it here. So this is going to be the cell body of a preganglionic parasympathetic nervous system neuron. And it's going to send its axon out, and this axon is going to go into the vagus nerve. So it's going to go into cranial nerve 10 here, which of course we know will emerge from between the inferior olive, and also the inferior cerebellar peduncle, so very close by. So it will just go into the cranial nerve number 10, and it will run down with the vagus all the way down to the heart. The vagus descends all the way down the body, so it's going to go by the heart. So these parasympathetic preganglionic neurons then, they can send their axons down with the vagus to arrive at the heart. So if I now draw the heart here, so let's say this is a cardiomyocyte, then what happens is once the axon of the preganglionic parasympathetic neuron has reached the heart, it's then going to synapse onto a postganglionic parasympathetic neuron at the heart, and you have very tiny little ganglia containing clustered postganglionic parasympathetic neuron cell bodies in the heart, specifically in the walls of the heart. So you're going to have these tiny little parasympathetic ganglia. Okay, so just completing up the picture then, here is the postganglionic parasympathetic neuron then. So this one is postganglionic, so I just want to now label up this picture to complete it. So this is the postganglionic parasympathetic nervous system neuron, and this will actually have its cell body in a tiny little ganglia in the wall of the heart. So I'll colour this one in red here. and this is the one which will then release acetylcholine onto the cardiomyocytes, and acetylcholine will have the effect of slowing down the heart rate by acting on the cardiomyocytes of the sinoatrial node and reducing the rate at which they fire off electrical signals by acting on M3 receptors, and on the contractile cardiomyocytes, acetylcholine again acting through M3 receptors, which is another G protein coupled receptor, so I'll just draw this on here. So here's my classical picture for a G protein coupled receptor. This is the muscarinic acetylcholine receptor type 3. The acetylcholine that's released by the postganglionic parasympathetic nervous system neuron will act on the M3 receptors, and this triggers cascades within the cells. And as I say, in sinoatrial node cells, the effect is to reduce the rate at which they fire off electrical signals, and in the contractile cardiomyocytes, the effect is to reduce contractility, reduce the force that a cardiomyocyte will actually contract with when it does contract. So when it receives the electrical signal to contract, it will now contract with a lower force, it will generate less force. Okay, just completing up the picture then, this one here is the preganglionic parasympathetic nervous system neuron. So this is the preganglionic PNS neuron here, like so, and I will just colour that one in yellow, although in this light it might not actually show up that I've coloured this in, but never mind. Here it is in yellow, if you can actually see that on the screen. Right, so overall then, in the Cardioinhibitory Centre, which is just this name for the combination of this portion of the dorsal vagal nucleus and the nucleus ambiguous, which contains these cell bodies of the preganglionic parasympathetic nervous system neurons, you have the cell bodies of preganglionic parasympathetic nervous system neurons, and these are going to send their axons into the vagus nerves, and the vagus nerve is going to allow these axons to get all the way down to the heart, deliver the axons to the heart, and then at the heart you have tiny little ganglia in the walls of the heart, which are the parasympathetic ganglia, where the preganglionic parasympathetic nervous system neuron will be synapsing onto a postganglionic parasympathetic nervous system neuron. So this can be one of these tiny little parasympathetic nervous system ganglions here. Okay, so this is a PNS ganglion, and there will be multiple synapses occurring in there, so multiple preganglionic parasympathetic nervous system neurons will be synapsing onto their postganglionic ones in this little cluster here, but they're tiny compared to the sympathetic ganglia in the sympathetic chains. And this is a general theme that you see all over the place in the parasympathetic nervous system, that the ganglia for the parasympathetic nervous system are generally much closer to the tissue that the parasympathetic nervous system is actually going to have an effect on, whereas of course the sympathetic ganglia might be very far away in these sympathetic chains from the actual tissue that they're going to act on. Okay, then the postganglionic parasympathetic nervous system neuron, it will then release the acetylcholine, so just connecting this all up into a flowchart, it's going to release the acetylcholine, which will act on M3 receptors on the cardiomyocytes, and the effect there is to slow the heart down, slow heart rate, and reduce contractility, so overall reducing cardiac output. So, the amount of stimulation then on the preganglionic parasympathetic nervous system neurons in the Cardioinhibitory Centre is going to determine the amount of parasympathetic nervous system output to the heart then. Okay, so let's now start to put this all together, so into the baroreceptor response. So, remember, the baroreceptive afferents, their central processes are stimulating neurons in the nucleus tractus solitarius. Okay, so the higher mean arterial pressure is, the faster you are getting action potentials coming up here. So let's say initially that mean arterial pressure is too high, and then we'll do what would happen if mean arterial pressure is too low. Rather than trying to do them both at the same time, let's just do them one by one. So what happens if mean arterial pressure is too high? Then the frequency of action potentials that are arriving down these baroreceptive afferents here, the central processes of them, is going to be too high. Okay, this is now going to be overstimulating the neurons in the nucleus tractus solitarius. Now, what are the neurons in the nucleus tractus solitarius going to do? Well, let's think about what we overall want the baroreceptor response to do. If mean arterial pressure is too high, then we need to lower mean arterial pressure. To do that, we need to lower cardiac output and lower total peripheral resistance. To do that, we need to increase parasympathetic nervous system and decrease the sympathetic nervous system, both to the heart and also to the vasculature. So we want to activate the Cardioinhibitory Centre, okay, which is in the form of the dorsal vagal nuclei and the nucleus ambiguous, and we want to inhibit the vasomotor centre and also the Cardioacceleratory Centre, and that's exactly what the nucleus tractus solitarius is going to do. The nucleus tractus solitarius is going to contain interneurons of both the inhibitory type and the activatory type. So you are going to have in the nucleus tractus solitarius activatory interneurons, which are going to go from the nucleus solitarius to the dorsal vagal nucleus, the Cardioinhibitory Centre in the dorsal vagal nucleus, and also to the nucleus ambiguous, like so. So these are activatory interneurons. So I'll just put this here. So those arrows, and I wish the picture was larger so I could actually draw the neurons, but unfortunately it's not. So I'll just draw arrows instead. So these arrows are representing activatory interneurons. Okay, so we'll have those coloured in pink. So the neurons of the nucleus tractus solitarius that are going to be activated by the central processes of the baroreceptive afferent, so these interneurons, you're going to have activatory interneurons and inhibitory interneurons. The activatory interneurons that have now been activated, and they're now going to fire action potentials more frequently as well because they're being stimulated more now, they're going to go to the nucleus ambiguous and to the dorsal vagal nucleus, and they're going to stimulate the Cardioinhibitory Centre. So now you're going to get increased activation of the preganglionic parasympathetic nervous system neurons, which means increased activation of the postganglionic parasympathetic nervous system neurons, which means an increased amount of acetylcholine released onto the heart, which is going to overall promote the heart slowing down and reducing contractility, i.e. lowering cardiac output. So that's how we activate the Cardioinhibitory Centre in the case that mean arterial pressure is too high. We have these activatory interneurons from the nucleus tractus solitarius to the Cardioinhibitory Centre. Meanwhile, of course, we're going to have inhibitory interneurons going from the nucleus tractus solitarius into the vasomotor centre and also the Cardioacceleratory Centre. So I'll have this shown here in blue with the blunt end rather than the arrow end here. So that in blue, that is representing inhibitory interneurons. Okay, so these are now going to be activated to fire at a higher rate because of the increased action potentials coming in from the central process of the baroreceptive afferents. So the inhibitory interneurons are now going to be firing at a higher rate, and that means they're going to be delivering more inhibition onto the neurons in the Cardioacceleratory Centre and also the vasomotor centre. So you're now going to get inhibition of the networks that are the vasomotor centre and the Cardioacceleratory Centre in the reticular formation of the medulla. And that overall is going to reduce the amount of firing that these output neurons are going to be doing. Okay, so overall inhibiting this network of neurons, which is the vasomotor centre or the Cardioacceleratory Centre, is going to result in fewer or a lower frequency of action potentials being sent down these output neurons, which then means that you're going to get less firing of the preganglionic sympathetic nervous system neurons and therefore less firing of the postganglionic sympathetic neurons. Okay, and that therefore means that the sympathetic stimulation to the heart is going to go down, and that's also going to help you bring down heart rate, bring down contractility, because remember, noradrenaline being released onto cardiomyocytes is it increases heart rate and it increases contractility through the Beta1 receptor. So just for completeness, I'll add this in. I didn't discuss this at the time, but just drawing another little picture of a cardiomyocyte here. The receptor on cardiomyocytes for noradrenaline is the Beta1 adrenoreceptor, and on sinoatrial node cardiomyocytes, activating this receptor by noradrenaline being released by the postganglionic sympathetic nervous system neurons is going to result in an increased frequency of electrical signal generation, and in a contractile cardiomyocyte it's going to result in an increased contractility. So reducing the noradrenaline that's being released onto the heart is going to overall reduce heart rate and also reduce contractility, so that will go hand in hand with increasing parasympathetic stimulation onto the heart to increase cardiac output. Okay, in addition, we'll also get reduced output from the output neurons of the vasomotor center, and that means that we're going to get less firing of the preganglionic sympathetic nervous system neurons and the postganglionic sympathetic nervous system neurons, and therefore we're also going to get reduced contraction of the vascular smooth muscle cells, so you're going to get dilatation of the arterioles in the skin, the skeletal muscle, and also in the gastrointestinal tract, and that's going to lead to a reduction in total peripheral resistance, which will also help to bring blood pressure down. So overall, that is the way that an increase in the firing frequency of these baroreceptive afferent central processes into the nucleus tractus solitarius is actually going to be converted into an increase in the parasympathetic nervous system output to the heart, a decrease in the sympathetic nervous system output to the heart, and a decrease in sympathetic nervous system output to the vascular smooth muscle cells around arterioles in these areas of the body that we've discussed. Okay, now let's discuss how it works in the opposite situation then, the mean arterial pressure going too low then. If mean arterial pressure goes too low, then that means that the frequency of action potentials that is firing along the central process of this baroreceptor afferent is going to fall. Now remember, at normal mean arterial pressure, there is a certain frequency of action potentials coming in here, so there's a certain stimulation to the nucleus tractus solitarius. If mean arterial pressure falls, then that stimulation to the nucleus tractus solitarius is going to go below what is normal. That means that the stimulation of these excitatory or activating interneurons is going to fall below what it would normally be, which means that the activation of the Cardioinhibitory Center that would be normal, that would be there all the time at normal mean arterial pressure, is going to go down. So you're going to reduce from normal the amounts that the nucleus tractus solitarius is stimulating these preganglionic parasympathetic neurons in the cardioinhibitory centers. Okay, and that means that their frequency of firing action potentials is going to go below what it would normally be, and therefore the activation of the postganglionic parasympathetic nervous system neurons is going to go below what it would normally be, and therefore the amount of acetylcholine you're going to release onto the heart will go below what it would normally be. So you usually are always releasing a little bit of acetylcholine onto the heart. That's known as parasympathetic nervous system tone. Now, because mean arterial pressure has gone down, you're going to reduce the amount of acetylcholine that's being reduced onto the heart below parasympathetic tone, which is therefore going to help bring up heart rate and bring up contractility, because you're now reducing parasympathetic tone. Okay, in addition, the reduction in the activation of the interneurons in the nucleus tractus solitarius means that now these inhibitory interneurons in blue here are also going to be firing less frequently than they would normally be firing if we were at normal mean arterial pressure. So that means that the inhibition that they usually provide to the vasomotor center and the Cardioinhibitory Center is going to go down. That means these centers go haywire, far beyond, their activity goes far beyond what it would normally be, because they've no longer got this brake on them that's normally present coming from the nucleus tractus solitarius, because those interneurons have now, they're no longer receiving their normal level of stimulation, and that means that the vasomotor center and the Cardioinhibitory Center are now going to fire more frequent action potentials down to the preganglionic sympathetic nervous system neurons, down these output neurons here. So you're going to get an increase in the activity of the preganglionic sympathetic nervous system neurons, and therefore an increase in the postganglionic sympathetic nervous system neurons, and therefore you're going to get increased sympathetic output to the heart, more noradrenaline onto the heart, which will increase heart rate and increase contractility. Okay, that will helps bring up cardiac output, and you're also going to get increased sympathetic nervous system output to the vascular smooth muscle cells, which will cause more contraction than would normally be there. And I should stress again, there's normally a certain amount of activation of the sympathetic nervous system to the heart and to the vascular smooth muscle cells, known as sympathetic tone, but now the amount of sympathetic output to these tissues is going to go beyond what it would normally be, beyond sympathetic tone, and we're therefore going to get a rise in cardiac output, and in the case of the vascular smooth muscle cells, vasoconstriction of these arterioles in the skin, gastrointestinal tract, and skeletal muscle, and therefore we're going to get an increase in total peripheral resistance and an increase in cardiac output, which will help us bring mean arterial pressure back up. So that then is the baroreceptor response, also sometimes called the baroreceptor reflex. The baroreceptor reflex specifically refers to how the heart rate changes in response to a change in blood pressure. But the full baroreceptor response also involves this change to the sympathetic tone to the vasculature as well. Okay, and this is the way in which we deal with a drop or a rise in blood pressure, and it is a feedback response rather than a feed-forward response. It's blood pressure has gone down, what are we going to now do to bring it back up? Or blood pressure has gone up, what are we going to do to now bring it back down? And it's the short-term response. We will have a break here, and in the next video we will begin the long-term response, which is the Renin-Angiotensin-Aldosterone System, and this is going to deal with the change in blood pressure by changing blood volume. So if blood pressure is too low, we're going to raise blood volume by the Renin-Angiotensin-Aldosterone System, and if blood pressure is too high, we're going to reduce blood volume by the Renin-Angiotensin-Aldosterone System. So we'll start that in the next video.